Hello, I'm going to be talking about transcription and translation. So here I have my DNA and RNA models. As you can see for this model, DNA has a double helix-like shape. And here's another DNA model to visualize the various components a little better. So here is the sugar phosphate backbone, and here are the nitrogen bases. There are 4 of them, which are adenine, cytosine, guanine, and thymine. Adenine pairs with thymine, and cytosine pairs with guanine, and vice versa. So the first step in this process is transcription, and there are 3 main steps when it comes to transcription, which are initiation, elongation, and termination. Initiation is when a section of DNA called the promoter instructs where and when transcription occurs. So let's say this is where it says transcription will occur. And then the RNA polymerase would unzip the 2 DNA strands. Then comes elongation, which is when an RNA strand is created and complements one of the DNA strands, which would be called the template strand. So let's say this is a template strand, and here's the RNA strand that would complement it. The RNA strand looks very identical to the non-template strand. However, one difference between these 2 strands is that thymine is replaced with another nitrogen base called uracil in the RNA strand. So that's the one difference to keep in mind between DNA and RNA nitrogen bases. And finally, there is a last step of transcription, which is termination, which signals when transcription stops. So at this step, the newly created RNA, which is currently attached to the template strand, would separate from that DNA strand. And after this, the RNA strand goes through a couple moderations, removing sections that will not be translated, and then the RNA is transported out of the nucleus to perform translation in the ribosomes to build codons and then make proteins. Codons are 3 nucleotides read from the RNA, to make specific amino acids. There are a total of 61 codons, I believe, that code for 1 out of the 20 possible amino acids. So protein synthesis begins when the codon AUG appears, which means to start the protein synthesis. After that, the codons create their corresponding protein until it reaches either UAA, UAG, or UGA, which all mean to stop translation. So this was my transcription, translation, modeling deep dive assignment. Thank you.